Hello, 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 everyone. Good afternoon, good morning, good evening, depending on where you're joining us from. It's very great to have you join us today, being day 2 of our precision medicine series for this year. And it would interest you to know that this is actually the first for this year. And what that means is that we plan to have other series this year specifically on precision medicine. So there's a lot to gain, there's a lot to learn, and I'm very happy and glad that the energy is so high that even about 30 minutes to the time, a lot of us have been able to join. Thank you so, so much. Thank you so, so much. So we're going to just talk about some housekeeping rules now before we delve into introductions and the chat box as usual. So, for those of us who were not in the last meeting, this is the second meeting we're going to be having on this series. And today we're very lucky and privileged to have a very, very, very great speaker in our midst who will be sharing with us some good news from the world of precision medicine and pharmacogenomics. Today's session is going to focus on pharmacogenomics, its implementation into clinical practice, and how we see the evidence in the field of pharmacogenomics, right? So at every moment in time, except when you've been invited to speak during questions and answer session, you're all encouraged to turn off your mic, to be unmuted, so that we do not have any interferences as much as possible. The meeting is designed to be able to give us great confidence, specifically this meeting, when it comes to pharmacogenomics and talking about evidence in the field of pharmacogenomics. So how has pharmacogenomics evolved over the years? What are the evidence that are available that we can say, okay, pharmacogenomics should be implemented into clinical practice? Those are the things we're going to be seeing today. And I urge every one of you to ensure that you stay glued to your screen. You might even want to have your notebooks and pen to jot down certain things. So that's for that. And so we'll be moving right now into the introduction session. So I'll just want us to go to the chat box, tell us your name, the country from which you're joining us, and your interest, why you are here today, why you've decided to be part of this session. For those of us joining from Nigeria, I understand that it's just, and some other parts of Africa, I understand that it's just afternoon. As a matter of fact, some of us would have been in class and all of those sort of things, but we've decided to be here today. So I want to see the beautiful messages about what your motivations are for joining us today. Thank you. So let's do that in the chat box while we wait for others. We're going to be starting as soon as it is 12 on the dot. That's in about um, 10 minutes from now. Thank you. Okay, seeing a very beautiful message in the chat box. Let's keep them coming in, guys. Let's keep them coming in. Keep them coming in. Okay, let's keep the messages coming in. Let's keep the introductions coming in. I want to see the place from which you're joining us, whether you're joining us from India, from Nigeria, from Gambia, from Togo, Ghana, Kenya. I want to see your messages in the chat box. Thank you. Okay, I see some beautiful messages in the chat box. Let's start with the first one we have here. I see, okay, Ikena Okori Darlington. Welcome, Ikena. It's great to have you here. You were even part of the last session we had, the last series we had. It's great to have you here. Your motivation is to gain insights on the relevance of pharmacogenomics in clinical settings. Certainly, that's something you're going to be able to gain a lot of insight on today. Thank you for joining us. I see Brian Omondi, biomedical science and technology students, Kisii University, Kenya. Your main aim is to gain more knowledge on relevance of pharmacogenomics in clinical care setting. That's amazing. It's great to know. Thank you for joining us. Christopher is joining us from Cyprus. Great to have you join us, Christopher. It's always a pleasure to see you join us. Thank you. I see Ben Onoja. Ben Onoja is a PhD vet medical student in UPM, Malaysia, and would like to know more about precision medicine. It's great to have you join us, Ben. Ben is very active. Ben has been with us, I think, since we started the series. That was in 2025 or thereabout. Ben has been with us, even during the last series. The very first one we had Dr. Luma. I think Ben was also with us. It's great to have you, Ben. Thank you. I see Asmwa from, I'm sorry if I mispronounced that, Mohammed. I see Mohammed, a pharmacy student from Nigeria. It's great to have you join us, Mohammed. I see Akinkemi, a medical student from Nigeria. It's great to have you join us today again, Akinkemi. You were here on the day one. That's very great to have you join us today. And you want to learn more about the relation of pharmacogenomics to clinical studies. That's really amazing. Okay, I see um, tennis, tennis. Um, I'm, I don't know if I'm pronouncing that correctly, from Pakistan, an undergraduate pharmacy student. It's great to have you join us, Tanis. Thank you so much. Okay, let's keep the messages coming in. Let's keep them coming in. Um, okay, I'm trying to see um, if our speaker is already in the house. Um, trying to see if our speaker is already in the house so we can begin immediately with the introduction, reading her bio, and then we'll get straight into the business for today. Okay, let's keep the introduction coming into the chat box. Just your name, for those of us who just joined us, your name, the country from which you're joining us, as well as your interest in pharmacogenomics and precision medicine. Like we said during the last session, what is the motivation behind your joining us today? There's a lot of factors, there's a lot of variables that come to play when we come to determine what you're joining us today would be. You could have decided to be somewhere else. You could have decided to be doing something entirely different from what you're doing now. You decided to buy data and all of those sort of things just to join us today. So we want to see your motivation in the chat box, your name, the country from which you're joining us, as well as your motivation for joining us. Let's keep them coming in. Let's keep them coming in. Okay. Okay. I see beautiful messages in the chat box. Let's keep them coming in. Let's keep them coming in. Okay. I see Esiosa Emanuela from D from Nigeria to gain more insight into precision medicine. Emanuela, welcome. It's great to have you join us. Um, let's keep the messages coming in. Um, I see, I see Aminata Sowe joining from Gambia, whose aim is to gain more knowledge of precision medicine. Aminata, thank you so much for joining us. I can assure you that before the end of this series, I'm sure we understand that we have even about 7 more sessions to go after today's session. We'll have 7 more sessions to go. So we have a lot of experts to talk to you about precision medicine. And I'm very certain, like I mentioned earlier, that at the end of the series, you would have gotten enough insight about what precision medicine is all about and its importance as far as clinical medicine is concerned. I see Faith, a pharmacy student from Madonna University, Nigeria, currently in class, but trying to be here. Wow, that's amazing. Faith, thank you so much for joining us. Thank you, thank you. I see Temitokbe Popola from Nigeria, aim to get and put knowledge gained into pharmacogenomics and use it as you work as a pharmacist. That's amazing. That's amazing. Let's keep them coming in, guys. Let's keep them coming in. I'm trying to look out for our speaker in the if she's in the waiting room or she's already joined us. So let's just keep our messages coming in. Okay, so I see Atere Mary joining from Nigeria, wants to broaden horizon, and you work as a dental technologist. Thank you so much, Mary, for joining us. It's great to have you join us. I see Kashmiri joining from South Africa. Thank you so much for joining us, Kashmiri. It's great to have you join us. Um, Kashmiri is a clinical pharmacist interested in implementing a pharmacogenomics program in that setting. That's amazing. Kashimi, that's amazing. Um, a clinical pharmacist, Kashimi, I don't know if you know about um, the project that has been going on in South Africa. I know that South Africa is probably the most advanced country in Africa when it comes to pharmacogenomics and precision medicine. There's a lot of work that has been done in South Africa and a lot of amazing experts there as well. Um, our speaker for day five, our speaker for day five is going to be from South Africa. Um, Nicole. Nicole is a very amazing pharmacist from South Africa, and she's done a lot of great work alongside so many other amazing experts in South Africa. So you probably want to link up with these people and be able to um, do more yourself. I see Eugene, a pharmacy student from University of Rwanda, interested in precision medicine. Welcome, Eugene. It's great to have you join us. It's amazing to have you join us. Okay. I see Ahmad Otaru from Nigeria, University of Abuja. Ahmad Otaru, great to have you join us today once again. I'm sure I read this name the last time. That was just some days ago. We had the first session. Thank you so much for joining us, Ahmad. I see Ife Jesu from Nigeria. Thank you for the series. Thank you so much, Ife Jesu. It's great to have you join us. Thank you, thank you, thank you. I see Chance Pacific. I'm not sure if I pronounced that correctly, from Rwanda. Thank you for joining us. Thank you for joining us. Thank you too, Chance. Thank you so much. I see Wesley from Kenya, interested in pharmacogenomics. Thank you so much for joining us. Okay, Kashmiri has heard of Nicole. Fantastic, fantastic. Yes, she's doing a very wonderful job in South Africa and alongside a lot of other amazing experts. So you want to link up with them as soon as possible. I see Sharon, a pharmacist from Kenya, really excited about the series. It's great to have you join us, Sharon. It's great to have you join us. It's great to have you join us. Okay, I see Oratile from South Africa, a pharmacy student. It's good to have you join us. Thank you so much. I see Dr. Omosola, a biochemist from Nigeria. Thank you to Dr. Omosola for joining us. Thank you so much. So again, I'm still trying to check for our speaker in the room. Let's keep the introduction coming in, guys. Let's keep them coming in. Just use the chat box. For those of us who just joined us, your name, the country from which you're joining us, and the reason for which you've joined us today, the motivation behind your joining us. Like I said earlier, there's probably a lot of other factors that could have prevented you from joining us today, but you've decided, regardless, to be here against all of those odds. So we want to see that reason in the chat box, what your motivation really is for deciding to be part of this session and this series as a whole. Thank you. Okay, I see even more messages in the chat room. Um, I see Miriam Oyinlola, a PharmD student from Nigeria. It's great to have you join us, Miriam. I see Paul Adeyemi from Nigeria, a fourth year medical student. Happy to be here. Thank you so much, Paul. We're happy to have you join us too. I see Chaka, a pharmacist and a DJ from Kenya. Thank you so much for joining us. You're excited to be here and you're happy to learn more about real world application of technology in healthcare. Okay, I see Shedrack. I see Shedrack from Nigeria, a pharmaceutical technologist. It's great to have you join us, Shedrack. Um, okay, Okay, so I was just finishing with our speaker now, so she'll be joining us like any moment from now. So we go straight to the business of the day, aside the introduction and all we've been doing. So let's keep the introduction coming in while we wait for her. I just finished talking to her now. Okay, so I see Stella from University of Nigeria, Nsukka. That must be Nsukka. Stella, it's great to have you join us. A pharmacy student, very interested in learning and broadening your knowledge on precision medicine. You are certainly doing the right thing, Stella. Precision medicine is here to stay, and it's the way to go. I see Adedapo from Nigeria. Adedapo, thank you so much for joining us. I see Azizat, biomedical engineering student in Nigeria. I'm sorry if I mispronounced that. You want to know more about incorporation of pharmacodynamics, okay, and precision medicine into biomedical engineering. Certainly, there are a lot of ways to do that, and I'm certain that um, even if you're not going to be able to get so much on that from this particular session, you'd be able to get more insight from subsequent sessions. I'm sure we have a session that is broadly on biomedical engineering, okay, that's going to deal into some aspects of biomedical engineering. So you don't want to miss that as well. It's great to have you join us. I see Miyoba, a pharmacist from Zambia. You want to learn more about the feasibility of incorporating pharmacogenomics into clinical care, especially in tight economies. Yeah, that's part of the things we've been talking about for years now. Yes, for years now, and it's part of the reasons why we're having this series in the first place. Besides, right? So we want to see how to um, incorporate pharmacogenomics into clinical care, and we've seen that at least studies have shown us that part of the challenges that we're having in implementing pharmacogenomics, especially in tough economies, like you mentioned, in tight economies, is because a lot of our people do not know about pharmacogenomics. A lot of the policy makers, a lot of the pharmacists. Yes, I think I gave an experience during the last session we had on with Dr. Zena about my experience with my superintendent pharmacist who probably has had 15, 20 years in the pharmacy practice, and he doesn't know about pharmacogenomics. Okay, so it's part of the reasons why we've organized this series to ensure that we get people to know more about pharmacogenomics. And with knowing more about pharmacogenomics, of course, you will certainly see how it benefits you and how it benefits your patients. So it's really amazing to have you join us in this call today, Miyoba. And then I see Eze Chika joining us from Ibadan. Thank you so much, Chika. It's great to have you join us. I think our speaker is already in the house, and we're just going to move straight to introducing her now, and we'll get to the business of the day. Thank you so much for joining us, Dr. Weam. Thank you so much. We are very grateful to have you
Thank
join
you.
us today.
Thank you.
Welcome, welcome,
Thank you very much.
welcome. So I'm going to move straight right now to introducing our speaker for today, and then we would move straight to the business of the day. Okay, so like we can already see projected on our screen, um, today is the second day of our precision medicine series. Part one, like I mentioned earlier, for this year, with the theme Precision Medicine in Practice, from Genomics to Real World Impact. And the topic for today's discussion is Pharmacogenomics in Clinical Care, from Evidence to Everyday Practice. And we have with us today no other person than Dr. Weam Aldiban, who is a pharmacogenomicist and a clinical pharmacy expert. So she's going to be taking us today through pharmacogenomics in clinical care from evidence to everyday practice. And Dr. Weam Aldiban is a lecturer and a researcher in pharmacogenetics and clinical pharmacy at the International University of Science and Technology. With expertise spanning pharmacogenomics, clinical pharmacology, and biostatistics, Dr. Aldiban has authored multiple publications in high-impact journals and actively contributes as a peer reviewer for leading scientific outlets. Her work focuses on genetic determinants of drug responses, personalized medicine, and advancing pharmacist education through academic leadership and entrepreneurial initiatives. Now, ladies and gentlemen, I want to see the amazing emojis in the house. I want to see your clapping in the room as we make welcome our guest speaker for today, Dr. Weam Aldiban. By the way, I hope I'm pronouncing that correctly, Dr. Aldiban. Thank you so much for joining us today.
Yes.
Fantastic.
That's correct.
The floor is yours now, Dr. Aldiban. Thank you so much.
Thank you. Thank you, Destiny, for welcoming. And I'm sorry for being late. I will share my screen now.
Absolutely. Let me stop my own now. Yeah. Yes, you can share your screen now, Dr. Aldiban.
It's okay now. It's okay now.
Not yet. It's not showing yet.
Okay. Host disabled content. Okay. I think you must give me an access so I can share my screen.
Okay.
Okay. I think now it's okay. Entire screen.
Okay. I'm not sure if
it's okay now.
Fantastic. Yes, it's okay now. It's showing now.
Okay. Okay. Is my presentation clear?
Okay. Dr. Aldiban, is it okay to put it in presentation mode so we can see it's yes.
I will share now.
Okay, I think it's still okay. I think we can go ahead, Dr. Aldiban. I just wanted to say if we could put it in presentation mode, like the slideshow on top, the slideshow that's showing on top. You can just click on the slideshow.
I need confirmation that my screen is sharing or not.
Yes, it's showing, Dr. Aldiban. It's showing. We can see it.
Okay. Good afternoon, everybody. Today we will talk about pharmacogenomics in clinical care from evidence to everyday practice. Pharmacogenomics or PGX is no longer futuristic concept or a poorly research-based discipline. It's increasingly becoming part of a routine clinical care across multiple healthcare settings, including hospitals, ambulatory clinics, and community pharmacies. However, translating pharmacogenomics knowledge into everyday prescribing decisions doesn't happen automatically. It requires intentional planning, a strong clinical evidence, interdisciplinary collaboration, and effective clinical decision support. So it's not easy to implement pharmacogenetics nowadays, but there are some steps we can step in to implement pharmacogenetics in clinical practice. So our session objects are they examine integration of pharmacogenomics into routine prescribing, review clinical evidences, understand implementation frameworks, and explore decision support tools in practice. Let's play this. Okay.
Yeah, fantastic.
Yes. Now let's step back and ask ourselves, why does pharmacogenomics matters in clinical practice today? First, patients receiving the same drug and dose often experience very different outcomes. Some achieve therapeutic benefits, others experience toxicity, and many fall somewhere in between. Genetics is a major contributor to this variability, and pharmacogenomics helps us understand and act on its individuality. Second, the burden of adverse drug reaction remains a major public health because they account for significant morbidity, hospitalization, and even mortality. Despite advances in therapeutics, ADRs are still among the leading cause of iatrogenic harm. And in another reason for the importance of pharmacogenetics lies in the limitation of trial and error prescribing, because our current prescribing paradigm often relies on trial and error. We start with a standard dose, monitor response, and adjust if needed. This approach is inefficient, costly, and frustrating for both patients and clinicians. More importantly, it delays optimal therapy. Finally, we can see that pharmacogenomics provides a framework to move beyond trial and error. By integrating genetic information, we can predict drug response, optimize dosing, and reduce ADRs. This is not theoretical. It's already being applied in oncology, psychiatry, cardiology, and beyond. So, what is pharmacogenetics or pharmacogenomics? They are the same, pharmacogenomics or pharmacogenetics. The definition is a study of how genetic variation affects drug response. This affects drug metabolism, drug transportation, and drug targets. To achieve the goal of precision medicine, the goal is giving the right drug to the right in the right dose to the right patient at the right time. So it's our goal to achieve precision medicine by relying on pharmacogenomics. Pharmacogenomics is a powerful tool for improving patient care, but it's important to understand that evidence alone doesn't automatically translate into practice. Just because we have strong scientific findings doesn't mean they will be used in clinics right away. In fact, history shows that there is often long delay between about 17 years between a discovery in research and its full adaptation in everyday medical practice. That's nearly 2 decades, where patients may not benefit from advances that could help them. So, this is why pharmacogenomics requires intentional implementation. It's not enough to have the science. We need the planning, infrastructure, and systems in places to make sure genetic insights are integrated into prescribing decision. Without that structure, pharmacogenomics risks staying in the research world instead of reaching patients who need it most. So now let's look at why this effort is so critical. The reality is that actionable pharmacogenomics variants are extremely common. In fact, large implementation programs show that more than 90% of patients carry at least one variant that can affect how they respond to medication. This means pharmacogenomics isn't just for rare cases, it's relevant to nearly every patient we encounter. Second, medications affected by pharmacogenomic variability are widely prescribed. Studies from diverse healthcare systems show that more than half of patients receive at least one pharmacogenomically actionable medication within a few years of follow-up. These include commonly used drugs such as clopidogrel, warfarin, antidepressants, codeine, and beta blockers, and statins. The third reason for why pharmacogenomics implementation is important is lying on the robust scientific evidence support clinical use for several drug gene pairs. This evidence supporting use relies on 3 pillars of evidence. So pharmacogenomic research has been ongoing since the 1950s, and for certain medications, the evidence threshold for clinical adoption has clearly been met. This evidence is summarized and operationalized in peer-reviewed guidelines such as SPIC, FDA drug labeling, and International Working Group recommendations. When we talk about the evidence supporting pharmacogenomics, it really rests on 3 strong pillars. The first pillar is robust genetic association data. We have extensive research showing clear links between specific genetic variants and how patients respond to medications. These associations are not just theoretical, they are backed by large studies across diverse populations. The second pillar is clinical guidelines and regulatory support. Organizations such as SPIC, as we said before, and DPWG have published detailed guidelines to help clinicians apply pharmacogenomic information in practice. In addition, regulatory agencies like the FDA have recognized the importance of pharmacogenomics by including pharmacogenomic information in drug labeling. Finally, the third pillar is demonstrated clinical benefits. Implementation studies show that using pharmacogenetics can improve patient outcomes, reduce adverse drug reactions, and guide more effective prescribing. This is the ultimate proof that pharmacogenomics isn't just about science, it's about making a real difference in patient care. Is my sound clear? Can you
Yes,
hear
yes,
me?
it's
Yes.
it's very clear,
Okay.
Dr. Aldiban.
Okay. So we have some examples of high impact drug gene pairs. As you know, for many drug gene pairs, there is strong and reproducible evidence linking genetic variations to drug exposure, efficacy, or toxicity. We have classic examples. These classic examples include CYP2C19 with clopidogrel, CYP2D6 with codeine and antidepressants, HLA-P star 5701 with abacavir, SLCO1B1 with statins, and the most famous example, CYP2C9 and VQRC1 with warfarin. So these associations are biologically plausible, clinically meaningful, and validated across multiple populations. So we have evidence for these genetic or gene drug pairs. Clinical guidance is readily available and continuously updated. The key resources include SPIC, which is the Clinical Pharmacogenetics Implementation Consortium, and FDA drug labels and biomarkers tables, Dutch Pharmacogenetics Working Group, DPWG, and Clean PGX, which is I think SPIC with GKP, as I remember, and peer-reviewed literature. These resources reduce uncertainty for clinicians and support standardized evidence-based decision-making. So we have a lot of benefits, as we said, of pharmacogenomics in routine care or pharmacogenomics implementation. These benefits are reduced adverse drug reactions, improved drug efficacy, faster time to optimal therapy, improved patient outcomes, and potential cost saving. Together, this data justify moving pharmacogenomics from optional consultation to routine clinical use. But there are some barriers against pharmacogenomics implementation. These barriers, like knowledge gaps among clinicians and patients, limited infrastructure and IT support, reimbursement challenges. By the way, reimbursement challenges, one of barriers, globally barriers, but limited infrastructure and IT supports, I think it's limited to low and middle income countries. Also, we have workflow disruption, evolving evidence for some drug gene pairs. We can say that not all evidence are reaching the evidence and SPIC. Some evidence are now evolving, like dabigatran and CS1. This drug gene pair evolving now. We don't have obvious guidelines for this gene drug pair. Not like warfarin, we have obvious clinical guidelines. So evolving evidence of some drug genes pairs is now continuous in pharmacogenomics. Although there are barriers, we have some drivers for pharmacogenomic adoption. So we have benefits, we have barriers, and we have drivers. The drivers are the key factors that push healthcare systems, clinicians, and policymakers to actually start using pharmacogenomics. Although there are barriers, these drivers are strong evidence for selected drug gene pairs, as we said before, warfarin CYP2C9, clopidogrel CYP2C19, and availability of clinical guidelines, advances in genotyping technology like NGS, decreasing testing costs, and leadership and pharmacists champions. All these factors are drivers for pharmacogenetics adoption. So we have, or we should have, framework for pharmacogenetics implementation. These frameworks for integration pharmacogenomics into routine care. Clinical implementation of pharmacogenomics is best understood as a three-phase process. So we have a three-phase process. First phase, planning, second, implementation, and the third phase is post-implementation. We will talk in details for each phase. The first phase, planning. The planning phase focuses on defining why and how pharmacogenomic program should be introduced. Then why? So why and how? Key steps include identifying the motivation for implementation, such as safety, maybe guidelines updates, maybe clinicians' demands. So we need to answer why. Then we need to justifying the program using institutional prescribing data, adverse event rates, and patient demographic. Then we must assessing available resources, including laboratory capabilities, informatics support, and personnel. Finally, we must determine whether the program will be clinical, research-based, or quality-improved focused. This phase ensures alignment with institutional priorities and realistic expectations. So this is for the first phase. Then we should move to the second phase, which is implementation. Implementation, the phase we will translate plans into practice. Central to this phase is the establishment of multidisciplinary pharmacogenomics oversight committee, which we will talk about it in details after minutes. So after establishing pharmacogenomics oversight committee, this committee will review evidence, select drug gene pairs, develop institutional protocols, design workflow, build decision support tools, then launch the program. So let's look inside this pharmacogenomics oversight committee. This committee includes pharmacists, physicians, laboratory experts, informatics, genetics, and administration. Their responsibilities are evidence review, protocol approval, education, and program governance. After that, we will move to the third phase, which is post-implementation. Post-implementation focuses on sustainability and continuous improvement. So the key activities include collecting outcomes metrics such as therapy changes, adverse drug events, and provider adoption rates. Continuing education for clinicians and patients, updating protocols based on new evidence or guidelines changes. Finally, expanding to additional drug gene pairs or populations. This phase ensures that pharmacogenomics remains a living, evolving component in clinical care. So, these are the three phases of implementation: planning, implementation, and post-implementation. We talk a lot about clinical decision support or CDS. CDS is essential for routine pharmacogenomics. Genotype data alone are not sufficient. Clinicians need conscious, actionable recommendations delivered at the right time. So, essential for routine pharmacogenomic use converts genetic data, which not sufficient, into actionable guidance, and finally delivered at point of describing. So what is the key features of, or what are the key features of pharmacogenomics CDS? Effective pharmacogenomics CDS systems integrate genetic results discretely into the electronic health record. First, trigger alerts when high-risk medications are prescribed. So, the first thing, it triggers alerts when high-risk medications are prescribed. Second, provide clear therapeutic recommendation and alternatives. Finally, allow clinicians to accept or override recommendations with documentation. I think it's something theoretical, and I will move now to some practical examples to be sure that everything is clear. Let's look at a practical example of how pharmacogenomics guides therapy. When clopidogrel is prescribed to a patient who carries a CYP2C19 loss of function genotype, clinical decision support system can step in. They may recommend an alternative antiplatelet therapy such as prasugrel. Importantly, this system doesn't just suggest a switch, it also provides supporting evidence and it checks for contraindications maybe, ensuring that the recommendation is safe and tolerated to the patient. This illustrates how pharmacogenomics can directly influence prescribing decision in real time. So clopidogrel and CYP2C19 is the most famous example for how CDS can help pharmacogenomics. As we said, we have many steps. First, identifies poor or intermediate metabolizer who carries star2 or star3 allele for CYP2C19. Then alerts the provider at order entry. Then recommends alternative antiplatelet therapy like prasugrel. Finally, supports evidence according to spec or according to FDA or according to EMA based decision making. We have another example now. This example is more complicated than clopidogrel. It's about beta blockers pharmacogenomics. Pharmacogenomics extend far beyond antiplatelet therapy. Another important area is cardiovascular medicine, particularly with beta blockers. Beta blockers are widely used, yet their effectiveness and tolerability can vary based on genetics. The CYB2D6, the enzyme plays a major role in metabolizing beta blockers. So, meaning patients with poor metabolizer status may experience higher drug level and more side effects, as we see on the screen. They may be suffer from bradycardia or hypotension. So, poor metabolizers may suffer from hypotension or bradycardia. On the other side, there's another enzyme, which is ADRB1, which is the target of beta blockers. Also, the variation in genotype of this enzyme can lead to variation in response to beta blockers. So we have two genes, not only gene playing a role or affect beta blockers. So in comparison with clopidogrel single gene, strong guideline-driven CDS. Beta blockers, multi-genes, we have two genes, evolving evidence, it's not strong evidence, it's evolving evidence, illustrates spectrum of pharmacogenomics implementation maturity, both relevant to routine cardiovascular care. So clopidogrel, obvious guidelines, one gene, but beta blockers, two genes, evolving guidelines. So as we move away from the one-size-fits-all approach to pharmaceutical therapy, we find ourselves at a crossroads between two primary models for delivering genetic insights, reactive and primitive testing. So we have two crossroads, and we will explain how these roads are different from each other. Reactive testing follows the traditional diagnostic workflow. A test is ordered at the time of prescription or after an unexplained adverse drug reactions has occurred. So the trigger, the adverse events. While in primitive testing, there's no trigger. In primitive, it's independent prescription stored for future. In other words, in primitive testing is emerging as a best practice because it ensures that actionable data is available at the moment a provider writes a prescription, effectively eliminating the waiting period associated with the reactive approach. So, reactive approach, as we said before, at time of prescribing, scope for this approach is single gene focus, like clopidogrel, CYB to C19. Whether the scope of primitive, multi-gene panels, whole genome, psychiatry, oncology, the consequences of this reactive maybe result in delays optimal therapy initiation. But in primitive testing, elements weighting covers broad variance spectrum. So the primitive framework is where we see the greatest impact on patient safety. Research shows that over 90% of individuals carry at least one actionable pharmacogenetics variant. The landmark PREPARE study validated this on a population scale and demonstrating that a 12-gene panel could reduce the incidence of clinical relevant ADRs by 30%. That in primitive approach. The economic case is particularly strong in specialists like psychiatry. Pharmacogenomics guided treatment has resulted in about 49 reduction in total treatment costs, primarily driven by 41 decrease in hospitalization. So these are the advantages of primitive approach. To move to or to look closer at reactive architecture, we see significant logistic hurdles. Standard laboratory-based reactive tests typically suffer from 5 to 7 business day delay. So when we order the test, we need to wait for about 5 or 7 days to get the result or to have a decision, which can critically stall the initiation of optimal therapy. In acute setting, like when we use clopidogrel, point of care testing has emerged as a specialized reactive niche. We see this in cardiology with CYP2C19 and for clopidogrel, or infections, disease like abacavir with HALA-P 57. These assays can provide results in under an hour. Yes, they are often limited in number of variants they cover and are not yet standard features in most clinical workflows. For us, as researchers and pharmacists, the most pressing issue with the reactive model is its single gene focus. So, single gene focus is the most problem with reactive approach. Everything is okay?
Yeah, absolutely.
Okay, because
Very well.
I can't see you on my screen, so I need to be sure. Okay.
Okay, absolutely.
Okay. Pharmacogenomics without advanced EHR systems. This is a slide for low and middle income countries like Syria, like my country, and maybe Nigeria and other countries. We are in Syria, don't have EHR system, advanced EHR system. So it's okay with pharmacogenetics or not. To be clear and to be real, still effective in outpatient and community settings. So all what we have mentioned depends on EHR, but what if we don't have EHR? If we don't have EHR or advanced EHR, manual CDS approach depends on pharmacists. On pharmacists consult notes at the first place. Standardized templates, we must standardize templates, and we must direct to provider communicate. We must have communicate with provider directly. So yes, it is still effective in outpatient and community settings if we don't have advanced EHR systems. So in hospitals, pharmacogenetics or pharmacogenomics programs may function as institution-wide pharmacogenomics services, specialty-based consult services, a strong integration with EHR and CDS, focus on safety and acute therapy decision. What about ambulatory and outpatient clinics? In outpatient care, pharmacogenomics is commonly implemented through pharmacogenomics clinics, medication therapy management, collaborative drug therapy management, and pharmacists play a central role in test ordering, in test interpretations, in patient counseling, and finally in provider communication. So the pharmacists play a critical role in ambulatory care in pharmacogenomics. What about community pharmacy? Community pharmacy, can we implement pharmacogenomics? Yes, we can. In pharmacogenomics practice models in community pharmacy, we need to patient education and engagement. So in pharmacy, in community pharmacy, the first step is patient education and get the patient or engage the patient in this education. Second, we can provide testing services. We can optimize therapy through prescriber collaboration with doctors, with other clinical providers. And finally, we can provide some manual or semi-automated decision support. It's not full automated decision support, maybe semi-automated decision support. So yes, we can implement pharmacogenomics in community pharmacy. So we come back to pharmacists and their role in pharmacogenomics. Pharmacists are uniquely positioned to lead pharmacogenomics implementation. Their role includes interpreting pharmacogenomics results, designing and managing CDS tools, educating providers and patients, both, conducting medications reviews, and monitoring outcomes and quality metrics. So we need to monitor outcomes. So we need to have some measuring success. As we move from theory into practice, it's important to ask, how do we know if pharmacogenomics is truly making an impact? Success in pharmacogenomics isn't just about running genetic tests, it's about demonstrating real value in patient care. Measuring success means looking at outcomes. These outcomes, clinical outcomes, like how much we can reduce adverse drug reaction, or how much we actually reduced adverse drug reaction by pharmacogenomics. How much we optimize therapy. And there are other measuring tools like operational metrics, like CDS usage, provider adoption, and finally we have another metrics like financial metrics. How many or how much money we are saved by implement pharmacogenomics. And the most important metric is reimbursement rates. This reimbursement rates is a critical metric in all the world, in developing and developed countries. Sorry. Okay. So, sustainability of pharmacogenomics program, how we can achieve sustainability for pharmacogenomics program. First, ongoing education. It's the most important things for both providers and patients. Leadership support, continuous data collection, integration into standards, workflows, and adaptation to new evidence. Adaptations to new evidence each evidence need, as we said, 17 years to be implemented. So which we are working today, we need about 10 to 20 years to be guidelines in pharmacogenomics. So it's hard work actually to do. Take-home messages. Pharmacogenomics is ready for routine clinical care. We have guidelines for many drugs. I didn't remember the accurate number, but there are dozens of drugs. We have guidelines for them. Evidence and guidelines support implementation. CDS enables point of care use, and pharmacists are central to success. Intentional implementation is essential. Intentional, not any implementation. It must be intentional implementation to pharmacogenomics. So, in conclusion, pharmacogenomics is rapidly transitioning from innovation to expectation in modern healthcare. The evidence is strong, the tools are available, and practical implementation frameworks exist. The key to success lies in intentional integration supported by clinical evidence, interdisciplinary collaboration, and effective decision support system. So that's the end. And
amazing. Thank you so much, Dr. Aldiban. I want to see the excitement in the house. I want to see the messages in the chat box. Thank you so much, Dr. Aldiban. It's been such an insightful session with you today. Thank you so
thank
much.
you. Thank
So,
you.
you're welcome. You're welcome. So it's time for Q&A for today's session.
Yes.
So in fact, personally for me, I had a lot of questions going through the session. So
Okay.
we're going to open room for Q&A now. So if you have a question, just put your hands up. You can use the icon here to just raise your hand and we will call on you, ask your question. If you won't be able to unmute, you can as well drop your question in the chat box so we can respond to them as soon as possible. So while waiting for questions from us, I don't see any hand raised currently. Um, Dr. Aldivan, I want to ask you a question that I think affects all of us. Countries in low and middle income countries in low and middle income areas of the world. And just December of 2025, the World Health Organization released a global genomic report about clinical research in pharmacogenomics and how it has evolved over the years. I think the study covers 1990 to 2024. And in that report, a lot were said, yes. But one of the things that stood out for me was the fact that the report identified a lot of disparities in clinical pharmacogenomics research, in genomic research generally. And about 80% of these studies are happening in high income countries with just less than 5% of the studies happening in low and middle income countries. And a lot of those times, you don't even have these countries leading the research. They are probably just providing samples or serving as secondary sites for the. And the report gave a lot of recommendations about how we could go about this, about ensuring that these disparities are broken down. But from your point of view and from how you see things in Syria and how you've seen things over the years in this part of the world, what do you think is that thing that we can do, even as pharmacy students, as pharmacists in our respective locations? During the introduction today, we got a lot of talks about, oh, I'm a pharmacist in Gambia, I'm a pharmacist in this part of the world, and I want to see how to implement pharmacogenomics in my setting. How do you think we could start bridging this gap, apart from the webinar series that we have, the educational forums that we have like this? How do you think we can actually start bridging this gap from your own point of view, Dr. Aldiban?
Yes. Yes. First of all, let's talk about the administrations, not about us like a pharmacist. Administrations of our institutional or institutions must or should take into account pharmacogenomics. As you said, about 80% of studies on pharmacogenomics are in developed countries. So what that mean? That means our countries are underrepresented as genotyping. Some genotypes are not in Caucasian, in Europe or USA. They are in our countries, in Middle East, in Africa, in South Asia. So we are underrepresented. We must collaborate with these global initiatives. First of all, to genotype our population. Some variants are very rare in USA or Europe, but they are very common in our countries. So when one drug release in USA, this drug will be tested in those population, not our population. So pharmacogenomics needs to be implemented, first of all, by collaborating with administrations or initiatives in developed countries. That's first step. Second step, we can rely on the must gene drug pair in our countries. For example, in Syria, we did many research. One of them about CYP2C19 and clopidogrel. Another, CYP2C9 and warfarin. The third one, CYP2D6 and tamoxifen. Our results demonstrated that CYP2C9 variants are very common. 2 and 3 are very common in Syrians, but CYP2D6 and 41 are not common in Syrians. So what we conclude from that? We will implement CYP2C9 and warfarin, but we won't implement CYP2D6 and tamoxifen because we are limited in resources. So we must identify what the gene drug pair we will focus on. I think it's the second step. And we need a lot of money which need, as I think, and as I said before, collaborative with initiatives and global initiatives to implement pharmacogenetics in our countries.
Amazing, amazing, amazing. Thank you so much, Dr. Aldiban. Thank you so much. Collaboration, the place of collaboration cannot be overemphasized. In fact, just to mention on this series, a lot of the things we've been able to achieve here at PharmaGenoXfE, which has been largely about creating awareness about pharmacogenomics, has been in a lot of ways inspired by the networks that I've been able to join myself, networks like PGRN, which I believe Dr. Aldiban is a member as well. PGRN, which is Pharmacogenomics Global Research Network, for those of us who do not know, is probably the biggest network of pharmacogenomics in the world. And they've been doing a lot in the field of pharmacogenomics. And so Dr. Aldiban has mentioned collaboration and how collaborating with global initiatives, global networks can really be very strategic to implementing pharmacogenomics in your country, in your region. So if you're here today in this meeting and you're not a member of Pharmacogenomics Global Research Network, you are not doing the right thing. I mean, if you want to go deep into pharmacogenomics and precision medicine, you should be a member of PGRN. And to make things even very simple, the best news is that joining them doesn't even require you paying anything. It's actually for free because they have the membership category that is specifically for people in developing countries. So you want to take that opportunity, just like Dr. Aldiban has mentioned now, collaboration, so that you can be able to stay on top pharmacogenomics in your region and be able to contribute to the field. Thank you so much. Thank you so much. So I'm still waiting for our hands. I do not see any hands raised. Am I not looking well? Do we not have questions for today for Dr. Aldiban? Okay, someone is saying, how can I join Okay, I'm not sure what you're asking. PARK. I'm not sure if that's what you're asking. Is it PGRN you're talking about? Chioma, could you unmute and say something? Hello? Yes, we can hear you. Go ahead. Okay, Chioma, your mic is not your network is not very strong. You probably would want to maybe leave and join again. Um, so just to assume that you're talking about PGRN, I'm assuming you're talking about PGRN. To join PGRN, it's very simple. Just head on to their website. You can just type PGRN on Google, on your Google search, PGRN, right? Just type it on your Google search and then you'll see their website. Just go to it and head to their membership section, and then you would see the other prompts that you should follow to join the network. If you still have difficulty, maybe after this session, you can reach out to me personally via the WhatsApp waiting room. So I'm assuming we do not have questions for today. So I'm going to be asking another question. Dr. Aldiban, can we still take more questions?
Of course.
Okay, great. So my other question is going to be about um, collaboration. You've mentioned collaboration as part of the ways, part of the means with which we could overcome the challenges that we're having as it relates to pharmacogenomics implementation, especially in LMICs. Now, from your own perspective and from all of the work you've been doing in Syria, what organizations or what networks, I'm probably thinking Pharmacogenomics Global Research Network, but I wouldn't jump to conclusion. What networks would you say have really been very strategic in helping you achieve most of the things you've achieved in Syria as it relates to pharmacogenomics? And also, what networks would you say have also assisted other persons in Syria or in other low and middle income countries with their journey in pharmacogenomics, especially with getting funding for research and all of those sort of supports?
Okay. Actually, we can try with the PGRN, as you said, and we can try with ACSPT. I will send you the link. They are interested in pharmacogenomics and translational science. But the most important thing, I think, and I will give you a hint, they are the companies, pharma companies. Like what? Like I think Roche and other companies, they are releasing some chips to do pharmacogenomics tests, and the companies of oncology. So if you have a research about pharmacogenomics, you can contact these companies and ask them for one thing: if they can help you do the test of sequencing, so you can genotype your population, and they will help because they have advanced laboratories, they have advanced equipment, so they can do the whole laboratory task with about 7 days, and they will have increase their popularity in your country maybe, and they will help you with your research. And finally we will add values to the pool of population. we have, as I mentioned before, we have some initiative in USA like All of Us. All of Us is about only US citizens. But what about Africa? What about Asia? So we can add value to this population if we send our samples and help and ask them to help us with the genotyping and implementation and interpretation. We can do it. We can do it because we have expertise and we can interrupt as our ADRs, our patients population, demographic, and etc. I hope this helpful for you.
Absolutely, absolutely, absolutely. Thank you so, so much, Dr. Aldiban. It's been an amazing time having you today join us for this series, and really grateful. We are really grateful. We want to say a very, very big thank you to you and to all of our participants today for joining us for this second day of our precision medicine series for the first half of this year. Thank you so much. So before we talk about signing the attendance for today, I'll just give some announcements and we would be done with our session for today. So the next session that we're going to be having in this series is going to be holding on the 7th of February. On the 7th of February is going to be the next session. And the time for that is going to be 7 p.m. GMT plus 1, 7 p.m. GMT plus 1. Okay, and we're going to be having a very amazing Professor Kazim Oshikoya, who's going to be taking us that day on another very exciting area of pharmacogenomics and precision medicine. So do not forget, our publicity materials will be going out, but just in case you want to mark your schedules, it's going to be on the 7th of February. And then before 7th of February, we're going to be having a live session on YouTube where we are going to be doing a lot of things. One of the things we're going to be doing on that day is to be having a mini tutorial and workshop. And the workshop is going to be basically on how to navigate CleanPGX websites. So if you've heard of PharmGKB before, you probably would have heard of PharmGKB. I think Dr. Aldiban mentioned that today, and Dr. Zaina mentioned it the first day we started this series. PharmGKB, which is now CleanPGX, is a tool that curates evidence from pharmacogenomics and guidelines, right? So we're going to be seeing how to navigate that resource. If you're a pharmacist, if you're a pharmacy student, you want to know how to navigate CleanPGX. So we're going to be having an expert on that day who's going to be taking us on how to navigate CleanPGX. And we're going to be having an Ask Me Anything session during that live streaming. And I've received a lot of questions about the Global Ambassadorship Program for PharmaGenoXfE, and we're going to be answering all of these questions during that session. And then we're going to be having a discussion which would be basically on emerging opportunities in pharmacogenomics and genomics in general, inspired by the World Health Organization Global Genomic Report 2025. So I spoke about the genomic reports released by the World Health Organization in December 2025 earlier, and we're going to be having a discussion on opportunities that you as a pharmacist, as a pharmacy student, as a biochemist, as a medical student, as a medical doctor, as a nurse, can delve into in your area, in your region, and in whatever field that you are. From this, this report highlights to us. So that's going to be happening on the 5th of February. So that's just 3 days from now. That's going to be happening on the 5th of February, and you're very welcome to join us. You do not have to register for that session. Registration is not required. Just do well to be on the ground, and it's going to be starting by 7 p.m. GMT plus 1. Stay tuned for that. And Just to emphasize again, a lot of persons reached out after the first series, the first session, about certificates, getting certificates for that session. Just to clarify, certificates will be given after the end of the series, the entire 9-session series. And each of us will be required to make at least 65% of attendance, right? So you want to ensure that you sign attendance after the meetings. If you're not able to sign attendance for any reason, maybe network, do well to reach out to me on WhatsApp. I'll do well to respond to you, even though it might take some time, but I'll certainly try my best to respond to you. Thank you so much. Thank you so much, everyone, for joining us. Do not forget again, the next session is on the 7th of February, 2026. That's in about some days from now. Once again, Dr. Aldiban, thank you so much. Thank you so much. We are very grateful to have you join us today. And to all of our participants, thank you so much. I don't know if you have anything else to tell us, Dr. Aldiban, before we call it a wrap for today's session.
Thank you. Thank you, Destiny. Thank you for this opportunity, and see you later.
Thank you so much. Thank you so much, Dr. Aldiban. Thank you so, so much. All right. Thank you so much, everyone. The attendance list is going in the chat box. I expect that we've seen it by now. Do well to sign the attendance, and it's a wrap for today. Thank you so much for joining us. Thank you so much for joining us.